did finish creating the exam. The numbers are posted in Moodle. Like I said, I left a column for multiple choice. I wasn't allowed to change the title of that. It wouldn't let me. So that still says exam 3, 100 points. Really, it's only out of 57. In Moodle, it says 60 because of that 3-point question that we're dropping, the one that didn't have answers to it, the leading lagging strand synthesis one. I did try to change the total in Moodle so the percentage would be accurate, but then it just made each question worth less, but there was no way for me to drop that question. I couldn't go in and say, don't count number 12 or whatever it was. So I put it back to 60 so that your numbers are the points you've earned, and that other one doesn't count. So don't look at your percentage on the multiple choice in the Moodle. Your points, if you got 57 out of 57, you got 100%. Right? If you got 40 out of 57, you could do the math. Right? And then so take your multiple choice raw score plus your written raw score, that's out of 100. So if your total is 86, you got an 86%. Okay, ignore the category percent for that one because I couldn't make it go away. Yes, ma'am. Sure. So remember when we talked about yesterday, right, that the last thing that we discussed was most genes are controlled at the level of turning on or off, which is transcription, right? And then after that, once they're on, then we can also control them by how long their poly-A tail is, how long they're there, post-transcriptional modifications, turning on and off. But most genes are just, we turn them on, we turn them off. That's how, at least in eukaryotes and in prokaryotes, that's how most gene regulation happens. So the most important level is transcription. Everything else is sort of fine-tuning, right? Ooh, let's make it last a long time, super long tail, short tail. Right. There are a few genes that are really controlled, their activity or the proteins controlled at post-translation, but they're much more rare. Where the proteins are always sitting around in our cell, we have to activate them by phosphorylation or decoupling with another protein or coupling with another protein. But that's very expensive energetically, so very few. Only things we have to have immediately. If we don't have them immediately active, the cell will die. Otherwise, if we have time to do transcription and translation, it's much more efficient to only have them expressed when they're actually needed. Okay, so the other ones are really just emergency methods. Okay, I do have the written part, which is sitting in my office. I forgot to bring them down. So if you want to run upstairs with me right after class and pick that up, you can have your written part back, or I'll bring them tomorrow class if you can last another day. But, you know, I'm happy. I meant to bring them down. I just forgot. Sorry. And then for the final exam, it's worth 150 points total. 100 points will be just like a regular midterm over this last week's stuff, right? It's the same amount of material, this last section, as for exam 3, right? So that's going to be 100 points. Just the same kind of questions, not comparing it to anything else, just sort of this stuff. And then 50 points are going to be the cumulative from exam 1, from exam 2, from exam 3. The only stuff from exam 4, or this last section on the cumulative, will be comparing back to some of the stuff you've already talked about. So, like, I'm not going to ask you a specific question about transcriptional regulation of the lac operon in those 50 points, but I might compare and contrast something about proteins involved or enzymes that we talk about to the enzyme chapter, right? Or compare transcription to replication. Replication was exam 3, transcription and translation is exam 4. So those kind of things, this stuff will show up in the 50, but, we're just doing it, so none of the specifics. But, big, important, specific stuff from exams 1, 2, and 3 in that 50 points. So again, it's only 50 points. We have 2 hours for the whole thing. There's not going to be more than maybe 30, 35 questions of cumulative, because we're going to run out of time if we do, 2 minutes per question or whatever. So it's going to be bigger stuff from each chapter. right? If you think about per lecture, during the course of this class, we've had about 25 or 26 lectures. So if you only get to have one question per lecture, what is it going to be? Or 2 questions per lecture, right? It's not going to be an enzyme we talked about once in one lecture and we never talked about again, right? It's going to be stuff that, you know, I would stamp my feet if you don't know it. Or Stein would be like, oh my God, the fuck are you people doing? Right, if you don't know about her vesicles or whatever. Okay, so the big thing's not a picky detail for the final. Okay, understanding the concepts, understanding big picture stuff, the common themes throughout the whole semester, all the processes, that kind of stuff. All right, we're behind. So we're going to talk about prokaryotic gene regulation and maybe a little bit of Ukes today. I did post a little transcription RNA processing tutorial summary of that lecture. I'll try to lecture capture some crap so we can do some question and answer stuff on Monday so we can get through all the material if you guys want to do that. So let's start. Again, these are sort of generic overall mechanism. Yeah. Yeah, we have 2 hours for the final. Probably. Yes. We'll just say yes. There'll be a written part on paper again, and then multiple choice. And I'll have a better idea of what hints for what that written might be. Not today. After I go think. Yeah. That's okay. The final part, if it's 50 points, and again, we're not going to have that much time because if we think about a regular midterm as an hour and 15 minutes, we really have like 45 minutes, yeah, for the final. So 25 or 30 questions for 50 points, so 1.5 to 2 per question, right? Less points per question than maybe a midterm. I don't know. Yeah, I don't know yet. It depends. I mean, because essentially almost all of replication was covered by the written, you know, I didn't need to ask that many questions in the multiple choice. And same with mitosis and meiosis. If you could draw all that, you pretty much know the answers. I asked about a couple of the phases. Right. So it just depends on what the written ends up being, how much it covers of the different processes for how many multiple choice, and how long I think it'll take, right? I didn't want the multiple choice to have 50 multiple choice, and then you guys having no time to do, you know, we had plenty of time for both. So that, yeah. So I just try to time it by how long in the past a written question like this has normally taken, and then I adjust the, you know, and there was genetics problems on the multiple choice, which take time too, which they're easy. You know what I mean? So I try to like make sure that there's enough time. I'd rather have more than less. Yeah. But right, then each one is worth more points. The more questions, yeah. And then, what was your first one? Was that the second one? Oh, the study guide question. I don't do study guides because in general, me typing out a study guide doesn't help you guys, right? The best way to learn material, don't roll your eyes at me, is to do it yourself. Passive learning is not passing. Looking at stuff, I went through my notes, that's not how we remember stuff, okay? And part of your job as a student is to determine what's important and what's not. What I talk about in class might be 1/3 of the stuff in the textbook, right? So that's already, my lectures are the study guide of what's important from the textbook. I don't pull sections out of the textbook that I don't cover. And not because I don't think they're important, but we can't cover everything. Yeah, so you can use the slides as your study guide, right? And use the book as your resource so you go back to the beginning and you're looking through a slide and you scribbled some notes, you can't read them, and you're like, I have no idea what Stein was talking about. I scribbled these notes and I don't remember, go back, read that section in the book. But you're not reading the book like I need to know everything in this chapter. So yeah, the slides themselves, if you just pull out all the stupid figures, those bullet points are a good study guide. But the best thing to do is when you're going through the slides, write down the 4 most important things from that lecture and make sure you know, and then make up some questions. Or Google some quiz questions or, test questions on those topics so that you focus on those. And we talked about topoisomerase. That's the only time we ever talked about it. It's unlikely to show up on the final. But compare, and what breaks hydrogen bonds, or what's the point of that, or, things that we've carried through. Polymerases, all polymerases do what? What's the difference between DNA polymerase and RNA polymerase? Those kind of things, are things that we talked about: transcription, replication, translation, complementary base pairing, anti-parallel, that kind of crap. The nucleotide stuff was in the beginning, but we keep talking about stupid phosphodiester bonds. Right, we're going to talk about peptide bonds. That's this stuff and from the beginning. So those kind of things will be what, yeah. And then the organelles chapter, right, there wasn't an overall Stein liked vesicles, so that you should probably make sure you know. But really, then we just randomly pick an organelle because, I would, for the final, I'm not going to have any written on the final. It'll be all multiple choice. I'm assuming hers will at least all be Moodle. So if there's fill in the blank or short, I doubt she'll do short answers. For the fourth, this last section, I'll have written on paper. No, we won't draw mitosis and meiosis again for the final, but you're likely to get a picture of a cell and ask something about a phase, or, what is this most likely to be in, or how many, 2n equals what if the cell looks like this and it's in G2 or whatever. Shit like that, yeah. Does that make sense? All right. Okay. So positive and negative regulation, we're talking about transcription, right? This is the on-off. Positive control in general is more likely to be used in eukaryotes, and negative control is more likely to be used in prokaryotes, at least the standard sort of classic ways we're going to teach you guys about gene regulation, okay? And the easiest way to remember is really, if it's negative control, the action that happens, which is proteins binding to DNA, right? Just like when we talked about turning on transcription, in general, sigma factor binds, RNA polymerase binds, that DNA protein interaction makes a process go, right? Then that RNA polymerase can start doing complementary base pairing. So if something binds to DNA and transcription is off, it's negatively regulated because the action is turning it off. It's a negative thing. Okay? If something binds DNA and it turns on, we call it positive. So just the basal transcription that we already talked about for prok, sigma binding, RNA polymerase binding, turning on, that would be positive. TBP binding, all its friends, and RNA polymerase binding, that would be positive. Yeah?
So for our case,
most eukaryotic stuff, especially what we're going to talk about in here, is pretty positive. Yeah. Prokaryotes can have both. Most things are controlled by negative, as it turns out. Okay? And so when we talk about turning on transcription with the sigma factor in the RNA polymerase or the TBP in the RNA polymerase, that's the basics what has to happen, minimally. There's other stuff going on that actually controls whether we get transcription or not. We sort of compartmentalize those so that we make it, this is really just what has to happen at the beginning. And then in these chapters, we talk about what else needs to happen to actually get a protein at the end, okay? And so, yeah, we always have to have sigma binding and RNA polymerase for a prokaryote, because if we don't have that, it doesn't matter what other proteins are binding. We have to have RNA polymerase docked to get complementary base pairing and enzymatic phosphodiester bonding. And in order to get RNA polymerase, we have to have sigma. Okay, so that happens across the board no matter what. If that doesn't happen, never get transcription. And same within eukaryotes. We get transcription if we get TBP binding and RNA polymerase, but in eucs we get a little bit, kind of physiologically irrelevant, not enough to do anything to change phenotype. Other stuff has to happen to get it to go on. And most of the time in eucs, it's forcing something on. Occasionally, it's shutting it off at the level of transcription. So most eucs is positive. All the examples I'm going to give essentially are mostly positive. The examples for proks are negative. Right? There are a few examples of positive regulation, but recent advances in the field, recent experiments done on those have already negated a few of the positively controlled operons. They're really not. They're really negative, and all the experiments that were done with their controls suggested, but as we get more sensitive experiments and better technology, right, we disprove hypotheses all the time. So positive control in prokaryotes seems to be less and less important, okay? And so that's just right. It doesn't mean those people were bad scientists and they were doing bad data. It just means under the circumstances with what they had to work with, that's how it looked like it worked, right? But hypotheses can be disproven, right, very easily with essentially one or two experiments. Okay, so that's the generic. And so if we were going to say, here's our gene, right, here's our promoter, right, and regulatory sequences, whatever, and if we show a protein binding and we get transcription, yes, what do we call this? Yeah. So that would be positively regulated, right? And so this is just any old protein. We're going to say that sigma and RNA polymerase, right, are always there. Something else had to bind to do something, okay? If something binds, right, we get binding of this protein to the DNA, and we get nothing, no transcription, sad face. What do we call that? Yeah, you guys are good. So when you have this question on the quiz or an exam, ta-da, 2 more points. Okay, so then these are just their pictures in the book of how they show that. Okay, negative control. If there's if it's a negatively controlled operon, and we really just have RNA polymerase, and we'll pretend this is prok, because it is, sigma and RNA polymerase, and no other proteins binding, we get transcription, right? Because all we need to get transcription alone is just sigma and RNA polymerase. The other stuff either can make transcription go faster, positive control, or something can bind and stop transcription. So for a negatively controlled operon or gene, right, if nothing's happening, nothing's binding, that means nothing's negative, except for sigma and RNA polymerase, those always have to bind. Then we get transcription. Only when we get binding, right, here's our guy binding, and we get nothing, we get sad face, we call it, right, that would be negative. So it would be a gene that's being kept off by that binding protein. Which means if we remove this, they're calling it a repressor protein because it represses. If we take this guy off, we should get transcription, right? Because we have RNA polymerase and sigma. He's not interfering with these guys binding. As it turns out, he's kind of sitting in front of it, acting like a bouncer. You can't get in the bar because the bouncer's standing in front of you and you're not on the list. Okay, so you're just sitting there. if you distract the bouncer, your friends can sneak in, right? Negatively regulated bar scene. Club, I don't know. I don't know what it is. I'm old. I haven't done it in a long time. Okay. And in positive control, right, we might have our RNA polymerase sigma sitting there, but it can't do anything alone. It needs to have an activator protein kicking its butt to get it to go forward and do transcription. This is very rare in proks. This is the one I'm saying, like, they used to think this happened a lot, and then they realized, oh, this little protein's there all the time. So it can't just be that if it's there whether it's on or off, right? More sensitive experiments really looking at what that molecular complex is. Yeah? I think when you're saying people are predisposed to a disease or predisposed to like some sort of cancer or higher risk, that's all about not about gene regulation, but actually the sequence of your genes, right? Predisposed to certain types of diseases can be based on your, not necessarily your blood type, but remember I talked about there's all the other proteins on your blood that in your immune system, right? So there are types of alleles that people have that are more correlated with autoimmune disease or diabetes or that kind of thing. And being sort of having higher risk for cancer is generally being born with a couple mutations already. So in general, cancer only shows up if, let's say you have to have 10 mutations in 10 specific genes. And I was born with 5 of those already, and you were born with 0 of them. My risk is much higher in my lifetime to gather the other 5 than you to get all 10. So not usually gene regulation, but occasional things like being lactose intolerant, that is at the level of gene regulation. Okay, and that's in eukaryotes, right? Anybody lactose intolerant? Yeah, you can't have dairy products because it makes, right? Talk about gas and bloating and pain, right? Yeah. Yes, that's what I'm going to talk about. This makes me so happy. Yeah, and the Lactose pills. So lactose intolerance just means you're no longer turning on the gene lactase, the enzyme that breaks up the lactose in your gut. And so as it turns out, all that sugar, right, lactose is a sugar, if it gets down into your intestines where all those bacteria are, right, which normally don't get to see sugar because you've already digested it up in your upper small intestine, it doesn't get down, doesn't get near your colon. Oh my God, talk about a population explosion of bacteria. There's this shitload of sugar for them to do glycolysis, right? And then they're making ATP up the wazoo, up your wazoo, as it turns out, right? Replicating like crazy, and in their metabolism, most bacteria give off CO2, methane, sulfate, stinky gas. So your cells do not make the gas in your gut, right? The only gas that is like ours is if you drink soda and, you know, the bubbles, you don't burp it out, some of that, or you swallow air. That's the only air that gets into your intestines that's actually from you. The rest of that air is all from your bacteria that live in your gut, okay? And so then all of that air and all that action and all the secretions of those bacteria give you that rumbling and the gas and the gas and the right? And so, right, lactose intolerance is if you stop making that enzyme. And so Lactose, the stuff you buy over the counter, is just that enzyme. So if you eat it with a little bit of lactose, that enzyme cleaves it to glucose and galactose, which then are easily put into glycolysis, and it never gets down to those bacteria to go apeshit. Yeah?
So kind of curious, I heard, well, they put it in association with like bacteria when they're in a certain environment, and there's like something present in some kind of factor, let's say lactose, and you say
right.
It would bind to what I assume would be the repressor protein, and then it would allow transcription to occur for that gene to express.
So that's in the actual bacteria.
That doesn't happen with
No, not in the ukes. So we're going to get to that exactly in this. The lac operon, how do bacteria, once they get it out of your gut, what do they do with it? But not our cells. So all of us, except maybe a very few, are born expressing that gene, right? There's very few babies that can't breastfeed and are allergic to the lactose in breast milk. Very few, right? Because especially like adults that are lactose intolerant, they were able to, some of them able to drink milk when they were kids, or drink milk as an infant, and then over time they can't. And so the hypothesis for how that happens is not even at the level of transcription, it's at the level of chromatin, which we talk a little bit more about in genetics. But it shuts down that gene so that you can't get TBT and RNA polymerase to get in there, right? And so evolutionarily, cultures that came from backgrounds where they domesticated animals and started using dairy products forever, right? The constant onslaught of having all of that lactose, right? Breastfeed till they were 4 or 5 years old, way back when. Oof, that sounds horrible. And then, you had a goat or a cow, and then you feed the kids milk because it's a year-round protein supply, right? You have a cow, you can feed your kid protein and fat, and much healthier than if you're just eating rice or grains or whatever, or waiting to catch a deer or something like that for protein, right? So cultures that were more likely to do that are less likely to become lactose intolerant as adults. Okay, so a lot of Asian countries, many people are lactose intolerant based on, right, they really only see lactose as infants. There's, again, no reason to waste our time making an enzyme in all of our intestinal cells that we don't use. So if the only time you ever see lactose is as an infant, it makes sense to turn that gene off and save efficiency. And so we turn those off, they think, by just squeezing down the chromatin so it never can happen again. And so sometimes people were fine as kids, mom made you drink a glass of milk every day, or the school lunch had milk every day, and then you go away to college and you're not buying milk and drinking milk, you're just, whatever you're doing, drinking other beverages. And then, you go to have a big fat piece of cheesecake, and then you're crapping your pants a couple hours later. Right? And so because you didn't have that lactose in your gut, those genes turned off. Right? Some people can not have milk for years and years and then have it and be fine. Right? They don't really know why it turns off in some people and not in others. But it does go with trends of your background. Did your ancestors thousands of years ago, not like grandma and grandpa, right, were they raised on dairy? Was it a culture that had dairy or not? And so then that sort of helps control it. But, you could just be some generic white person from Europe, which most people are not lactose intolerant, and then, you turn 49 and you have a piece of cheesecake and you shit your pants. You're like, what the hell? I was fine 3 weeks ago drinking a glass of milk, right? So sometimes it can be, and we don't know how that happens exactly. Yeah. Right.
But were we ever supposed to be able just to drink?
Well, so one of the hypotheses, I love talking about this shit. One of the hypotheses through evolution was, right, when we were Australopithecus, right, we were more like apes than like people, right, more like Lucy, right, and so they weren't brilliant, but I don't know how we survived really. I know, and how wimpy people are, in general, compared to those people were tough. Holy crap. Anyways, so they think the transition between the frontal lobe and having our reasoning and what makes us people, the thinking, the reasoning, to be able to plan ahead, to learn, to make choices, correlates with when our ancestors started hunting for meat and meat protein and animal protein. And so the difference between lineages that died off and lineages that continued and then split were, one of the hypotheses was those were the lineages that ate meat and had animal protein, allowed us to gain more weight, have extra fat. right? Use more energy to dump into our brains rather than just survival. And so the, transition from those guys to the Homo erectus, habilis, all of those other guys, the Neanderthals, right? Those guys were tough, right? But we were smarter, as it turns out. Well, we had a bigger frontal lobe. We're guessing we're smarter. But they were tougher. So really, they should have won. Like, that was harsh situations back there. But I guess, I don't know. We just had sex with them. Took all their muscle. Yeah. See this brow line right there? It's pretty Neanderthal to me, guys. I don't know. Get this jaw right there. But my chin, that's all Homo sapien. So yeah. So were we supposed to? Evolutionarily, it gave people that drank milk that had like this year-round supply, right? Domesticating animals went back way, way, way back. Deer, whatever, catch one, keep it around, milk it. Now your kids can have fat and protein every day, rather than once every 3 weeks when we get that deer, right? The rest of the time we're hunter-gatherer, nuts and berries and sticks, right? And so that gave them a huge advantage, right? And then evolutionarily, our physiology has adapted to carbs, fats, proteins, right? Our teeth are totally carnivore, but still some herbivore. So, whether we are meant to or not, through the course of getting here, all of those different things helped us. Whether they're good for us now when we have calories up the wazoo, we're not on the brink of starvation all the time, that's then nutrition. And we want to live to 100, right? Cavemen maybe got to 30, so none of the old age stuff had anything to do with natural selection because it didn't exist. There was no menopause. You didn't get old enough to hit menopause, right? And menopause, if it hurts women, who cares? They already had babies. It doesn't affect passing of traits, right?
Certain things our bodies are starting to reject as time goes on, and could that be attributed to the fact that we don't really need it?
Anymore, probably, right? We don't need extra calories, at least not in the United States, right? Developing countries where there still are people starving and they are malnourished, then, but yeah, we got cupcakes, we got fat galore, right? We don't have to, nutrition-wise, that's a whole argument of whether milk is, the good outweigh the fat, blah, 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 and that kind of stuff. All right, sorry, we digressed. All right, so we already did that. Okay, so now we're going to talk about what do bacteria do. So that's what our gut does with lactose, right? So the bacteria in our gut, the E. coli living in your colon or in your small intestine, right, prefer to use glucose whenever possible. And if we think back to glycolysis chapter 8 or 9, whatever that was, right, our cells like glucose too. That's the easiest molecule to send down glycolysis, right, pyruvate processing, Krebs cycle, electron transport, shitload of ATP, right? That's the easiest molecule, okay? Any of the sugars that are disaccharides, first we have to cleave those. Most of them have at least one molecule of glucose in it. If it's galactose, we have to modify that, which then makes it glucose and send it, there's stuff we have to do with sucrose, lactose, all those other things. So glucose is the easiest fuel. Right? So if you're a marathon runner and you're trying to win your race, right, you're going to eat that goo shit while you're running or drink that stuff, which is glucose, not sucrose. You don't want table sugar in there because your body has to cleave it first and then send it. You want that to jam out of your stomach or your intestine into your bloodstream as glucose sucked into your muscle cells, boom, right, go to your electron transport chain. Right? That's the best thing. If you're trying to lose weight, don't do that. Don't work out really hard and then guzzle a big Gatorade. It's got like 50 grams of sugar in it. Right? What was Gatorade made for? Football players. The Florida Gators coach formulated this because they're out there for 8 hours in the hot sun, sweating and running. Do football players want to lose weight? What do they want to do? Gain weight. Drink that unless you're trying to gain weight. Okay? So we don't want all that sugar. So bacteria, glycolysis is the same, essentially, right, in bacteria as in eukaryotic cells. Same kind of process, right, except what do bacteria do? Do they have a Krebs cycle and electron transport chain? No. How can they? Where does that happen? Yeah, that happens in the mitochondria. They have similar stuff. They use other membranes, blah, blah, blah. But if we're just talking about glycolysis, using glucose, right, getting to pyruvate, then they can either do fermentation to make alcohol, yay, ethanol, or, some of the boring ones still make lactic acid, right? Bacteria that make yogurt, they ferment lactic acid. That's that sour taste in yogurt, okay? So they really want glucose. They can use other stuff when glucose is not available, like lactose, but they prefer glucose, so they'll only use something else when there's no glucose available. All right, we're going to skip this because I talked way too much. That was just showing how they know it won't use lactose if glucose is available. Okay, if both are available, still just uses glucose. Okay, because always glucose first, same with us. Okay. So E. coli, or the bacteria, the prokaryotes, make an enzyme that breaks down lactose only when lactose is available. So it's efficient. They don't make this enzyme unless it's useful. Okay, so that's at the level of transcription. It's off if there's no lactose around, or if there's any glucose there. It only turns on when lactose is inside that bacterial cell and no glucose is around. Okay? So that tells you that the presence of lactose in that bacteria induces gene expression. Okay? Whether directly or indirectly, we don't know yet, but, right, you put lactose in that cell, no glucose, boom, we turn on gene expression. So we call that the inducer, right? To induce something is to make something happen. A molecule that stimulates expression of a specific gene. So that's the lactose. The question is, does that sugar itself directly bind DNA, directly do something to... That's the question. How is it the inducer? So when they first looked, if it's there, we get gene expression. If it's not, we don't. And so they figured out the lactose metabolism operon, and they use that as a classic example of how bacterial genes work. Okay? And one of the things we want to talk about is in bacteria, we have, they're not showing us here, right? We always have a promoter region, and remember we talked about the -10 and the -35? So those would be within the promoter region. And then in bacteria, we can have multiple genes sitting in a row, and we make one big RNA of multiple genes in a row. Okay, so this would really be, well, it's actually 3 different genes. They sit all together. They have one promoter, one mRNA. When they're translated into 3 different proteins by where, all the docking of the ribosome. But they can be all together one big RNA. In eukaryotes, this never happens. Okay, in eukaryotes, we have the promoter and a gene, promoter, one gene, promoter, one gene. We never have multiple genes in a row that have the same RNA. So every piece of DNA has its own little piece of RNA and its own promoter. Okay. In prokaryotes, we put them together, and the ones that are together under one promoter make sense because you either need all 3 or none of them. And so all 3 of the genes in this operon are all about metabolizing lactose, some aspect of metabolizing lactose. So the cell, right, it's a single cell. It either needs all 3 because lactose is there and we want to break it down for glycolysis, or no lactose is there and we don't want any of them. So it would be a waste of energy and space, because think about bacteria have a little chromosome, to have each gene have its own promoter if they're always on together and always off together. So it's nice. We have one promoter, 3 genes. They're all on or they're all off. So it's much more efficient. Whereas we have 46 chromosomes that are ginormous, and we have multiple tissue types, right? These 3 genes, we might need all of them in one cell, but only 2 of them in another, and 1 in another. So everybody has their own promoter. Everything is regulated separately. In bacteria, lots of genes are together. Set of 3, all on, all off. Set of 4, 1 promoter, all on, all off. And so they're just showing us, right, this little piece of DNA out here is on the bacterial chromosome, and the process of breaking down lactose. First it has to get in the cell, right, the beta-galactosidase enzyme cleaves it into glucose and galactose. Now we can put both of these guys into glycolysis, right? So part of it's getting it in the cell, breaking it up, and then there's another gene that does something else. Okay. So this is all 3 of those that I talked about are transcribed together. Coordinately regulated means all on or off. Right, that's all. There's one other gene that's involved in this, right? When we look at if these are all on or all off, we know sigma has to bind an RNA polymerase, and those are almost always available. So how do we control whether it's on or off? We need to do either a positive regulation or a negative regulation. And so what they found was this other gene that happens to sit upstream that makes another protein that acts as a repressor. Okay? Oh, crap. Here we go. So this is really how it's set up. This would be the lactose metabolism genes. Okay. Here is the promoter. And then there's a sequence that sits right next to the promoter, between the promoter and the actual genes, that we call an operator. Operators only exist in prokaryotes. Operons only exist in prokaryotes. So if you see or hear either of those 2 terms, has to be prokaryotic. Promoters, both, right? Proks and eucs have promoters. No operons in eucs, no operators in eucs. Okay, only in proks. And so we know, or I've been telling you, that RNA polymerase and sigma, right, we'll put our little sigma guy in there, can bind to the promoter on the bacterial chromosome pretty much all the time. Okay? And if that happens, then we'd have gene expression of everything. Transcription would be on everywhere. We know that'd be stupid, because that'd be a waste of energy to have every bacterial gene on, just hoping it's needed. And you've already been told that the lac operon or the lactose metabolism genes are only on if lactose is there. That means when there's no lactose, right, and you just ate a big Snickers bar, and it's just glucose and sucrose, right, your bacteria are just going to be using glucose. So they want this to be off. So how it works is there's another gene up here that has its own promoter, and this one's always on. There's no regulation to it. It's just on all the time. When something's on all the time, we call it constitutive or constitutively active, always on. So we always are making this protein, always present in the cell. Okay? If it's naked, if it has nothing attached to it, if it's just happy-go-lucky protein that, right, we transcribe, we translate, make this protein, that guy binds to the operator. Okay? When it binds to the operator, it's the bouncer. It's physically sitting in the way of sigma and RNA polymerase from passing through to do transcription. Right? This is where we'd want to start complementary base pairing over here. Right? RNA polymerase is stuck right there. If it can't get to here, it can't do anything. So the repressor protein is actually blocking access to where it wants to start doing transcription. Yes? That's a great question. The operator is the sequence of DNA. Great question. The repressor is the protein that binds that sequence. Okay, does that make sense? So the operator is more like a promoter. The promoter is the sequence. Sigma and RNA polymerase bind to it are the protein. The operator is the sequence. The repressor protein binds to it and blocks access of the polymerase. Cannot get transcription because it's blocking access. Okay? The only way we can get transcription is if we remove that repressor or get rid of it. But it's always on over here, so there's no way to turn off that gene. We're stuck with that protein all the time. So we have to do something to that protein so it can't bind. Okay? Because we're stuck with it always on. Right? There's no option to try to turn off that gene. Always on. Right? So here they're showing us the lac. Right? This guy's always doing transcription, always doing translation to make the protein. If that happens, it's always binding here. Right? We'll make our sigma guy and RNA polymerase sitting there. Okay? If that's the case, then we don't get any transcription. Right? Sad case. Yeah. Yeah, so again, we're always lying. In any case where things are binding, it's always kind of an on-off, right? It's not like a pit bull that grabs and bites and doesn't let go till you suffocate it, right? It's like a regular dog. Ooh, bit you. Ooh, bit you again. Oh, bit you again, right? And so it's just doing on-off, but a snapshot in time, it's mostly on, it's doing its job. So that's why it can get pulled off, which we'll get to that in a second. So yeah, if there's nothing else going on, if there's glucose around, this is what happens, right? We don't want the lactose metabolism genes expressed if we don't need them. If glucose is there, we're making the stuff that breaks down to send glucose through glycolysis. We don't need this stuff, okay? Right? If we don't need it, we don't want it on, this is what it looks like. If we've used up all the glucose, and now you just drank a ginormous glass of milk, or a big old piece of cheesecake. Cheesecake. Now there's lactose around. I'm just going to make it like that. Isn't that a good piece of lactose? It's actually, it's a disaccharide. I guess I should make it like that. Okay? That lactose has an affinity, or is in love with, the repressor protein. Okay? So when lactose is around, blah, 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 it will stick itself to the repressor protein. And now remember, we're making lots of it. There's free repressor protein all over the place, right? We're transcription, translation, and so all of our repressor proteins are going to attach to a lactose. There's conformational change, as Dr. Stein walks in. Conformational change so that, right, this guy used to have teeth to grab the DNA, and now when lactose binds to it, it's like, he lost his dentures. So he can no longer bind to the DNA if it's attached. Got it? Got it? Like that. So the lactose you can think of as sequestering all the free stuff and pulling off the guys that were stuck to the DNA. Well, look. Oh my God. RNA polymerase is so happy because the bouncer's gone. What happened? Oh, come on. Get more excited than that, people. Work with me. Woohoo! Come on. Here, let's make it purple. Yay! Woo! Transcription and translation can happen, right? Let's pretend like, here, let's draw teeth on this guy. right? These are the proteins that are involved in breaking down lactose. Now, these guys can come over here and go, yummy, and break up lactose, to glucose and galactose. Now we can use those to do glycolysis. So they're going to run around, doing that to all these guys. What happens when now we set free some of these repressor proteins? Oh my God. And then what happens to transcription? Holy crap. Right? And then we're still making more repressor protein because that never stops. Right? And so we're bound, blocking, no more. Right? We've used up all the lactose, we've broken it all down, now we don't need them anymore, it turns back off. We did that discussion already. I already talked about crapping my pants, or your pants, or their pants. Whoops, I've crapped your pants. I know. It's true. Okay, so this is the slide of what happens, right? Lactose is absent, right? Which means the repressor can bind, no transcription. Lactose is present, right? Pulls it off. This is like a ghost of it. They shouldn't even have had it there. They need it. So, right? This guy can just buzz through and do transcription. And then this one is, if we had a problem and a mutation in that gene, right? Let's say there was just some mistake that happened during replication, mutated that, we don't make, we make a protein that doesn't have teeth, or we don't make the protein at all. If there's never a repressor, then of course we're always going to get that to go, right? Yes?
How long will the testosterone constantly making nothing but repressor, and when you keep that from overwhelming and binding, that's right away?
It's however, right, when there's a huge amount of lactose, it seems to suck those up. I don't know what the half-life of those proteins are, right, when they're ubiquitinized and broken down. Yeah, so I'm assuming it probably has a relatively short, because otherwise, yeah, if it's just constitutive and then those proteins never break down, you could never get enough lactose to suck them all up and stop that. So they got to have some sort of, turnover, right, so that it's efficient. And it seems to be efficient, so they must not stick around that long. And same with the RNA, must not stick around, must not stick around that long either. Okay. Okay. All right, so we're not going to talk about this too much, but originally we thought, or scientists thought, that there was also positive regulation for the lac operon, because situations where both glucose and lactose are there in the media that we're growing the cells in, like in your gut or in a flask, we still don't see any genes for lactose metabolism. And that doesn't make sense if we're thinking, well, if lactose is there, we don't get binding, we should always go. Why is the presence of glucose, how does that block this? Right? It's easy to see lactose only, right? We get transcription because it's binding to the repressor and pulling it off. Glucose only, well, there's no lactose, the repressor's always binding. But what about when both are there? And so they came up with this ridiculously long scheme of positive regulation where something binds and blah, 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 only if glucose is there, so forth and so on. And then recently, within the last couple of years, they realized, oh, that protein's always there. So it can't be positively regulated by something that's always there, whether it's on or off. And so what they've discovered by being more sensitive is that if there's glucose and lactose in the media or in your gut outside the cell, only glucose can get in. The only way lactose gets in is if there's no glucose around. So there's never a place where inside the bacterial cell both are available. It's always one or the other. So what we learned is always true, because there's never glucose and lactose inside the cell at the same time. So we don't even need to worry about that. So entire chapters in all these books have been cut because that was half of this chapter was all about the positive regulation, which is totally not true. So again, science is always progressing, and if I don't read the new editions and it's not my field, I might be teaching wrong for years on end. Yeah.
So then it comes down to stacking transfer between both glucose and lactose.
Yes, coming through. Yep, coming through the membrane. Exactly. So that was this. This is inducer exclusion. Blah. I don't care about the name of that or whatever. But there is no case where they're both in there. So really, we're just going to learn the negative regulation of lac operon, just what we talked about.
So do they both go through the same channel?
No, I don't think so, right? I think what they're saying is if there's glucose out there, it blocks this. I don't know, I'd have to read the paper. I did go and look at the primary research because when I read the new edition, I didn't really buy it. I wanted to make sure, but I don't know. Yeah, no glucose and lactose are ever in there at the same time. So the cam, all that is bullshit. Yeah, well, it's a good thing because it's bullshit anyway. Okay. Okay, so then these are just talking about some of the mutants, right? If you mutate this channel protein, and it doesn't work, it doesn't channel anything, what would happen? No, well, you're dead. No lactose will get in, right? So then we're never going to turn on the lac operon, right? The repressor is always going to be bound because there's no lactose available.
And so is this true? So he's teaching me this. So you said that this is the way most bacterial proteins work. They're always off.
Almost always, yeah. They're either constitutively on all the time, unregulated, or they're held off. Eukaryotes are mostly the opposite, yeah. There's a couple examples of positive regulation that I do in 360 when we get to it, but they kind of... I think it's just simpler in a bacteria where there's not so many genes and there's not so much going on. You have one cell, you either need it on or off. There's no like, oh, in this liver cell I need it on, but in the liver type next to me I need it off, right? Because in eukaryotes, we hold things off by chromatin, so there's lots of holding off, but we don't call it transcriptional regulation because there's no opportunity for transcription. That's the whole epigenetics crap. So eukaryotes do hold stuff off, lots of stuff off, but not at the level of transcription. They hold it at the level of chromatin, which that's all 360 stuff. Yeah. Oh my God, we didn't do any uploads. Okay, so if you have a picture, draw a smiley face or something, and or somebody with diarrhea and eating cheesecake, and then take a screenshot and upload. Yeah, but it'll just say late. That's fine. Or a selfie, yes. Dude, make a drawing,